We started in 92 and it is then after I moved Actually, we are
So good morning, everyone. So we are going to start. So as usual, people online, do not hesitate to report any technical issue in the chat, and please ask your question rather in the Q&A tab, and we're going to allow you to interact with the speaker at the end during the discussion session. And with that, thanks very much, Aline, for introducing Brandon today.
Oh, thanks for all the organization of the seminar, the committee, and thank you all for coming. It is our pleasure today to host Brandon Gaut. So Brandon is a distinguished professor at the University of California, Irvine, and he has done really varied work, a lot on domesticated plants. Also, has also worked on experimental evolution in bacteria, also in plants, and he's focusing lately on the grape, Vitis genus, and it will be the focus of his talk today. Yeah, so I wanted to mention Brandon is currently the editor-in-chief of Molecular Biology and Evolution, MBE, just in case you didn't know. And yes, so today he will focus on climate change and adaptation of grape vine to climate change. So thanks, Brandon, for coming, and I'm looking forward to your seminar.
Well, thank you. I feel like as an evolutionary biologist, you really haven't arrived until you've spoken at Montpellier. So I'm very grateful to be here today, and I appreciate Aline's invitation and the committee and all of you who are here. So thank you for coming. You may notice that those are not grapes in that photo, but I thought it might be fitting to show you olive trees by Pont du Gard. It's right. Do I say that properly? The Roman, and so we're starting with a bit of a French flair here in this. But I will be talking about grapes, a little bit about the domesticated grape, and then even more about the wild relatives of grapes in the genus Vitis. So as Aline said, I sort of started my career studying the process of domestication from a population genetics standpoint. And it was back in the day, and it was actually early days for doing these kind of coalescent models, and that's a model on the left, and trying to model what in population genetics terms domestication looked like. And the take home I want you to get, don't worry too much about that figure, is that when we studied maize, that's what I started with, we found that basically there had been a very severe bottleneck. And that makes sense when you think of domestication. Domesticators are going out into the wild, choosing some subset of that wild diversity, and then domesticating it. Great. So I stopped working on domestication many years ago, but then I ran across this really interesting, that there is very little evidence for a bottleneck effect in perennial crops. In what annual crops, you see that all the time. There's this very strong bottleneck. And so it's kind of puzzled me. And so perennial fruit crops have 95% of the genetic diversity found in wild populations on average. And I read that actually in a paper from 2011, Miller and Gross. It's a really wonderful paper if you have the chance to read it. So I thought that's really puzzling to me. How can you domesticate something? After all, domesticators are taking a subset of the genetic diversity. Why don't you see this decline in genetic diversity in perennial crops that you see in annual crops? So I thought I should start working on perennial crops. And so I said to myself, what would I work on? Well, this is my favorite all-time crop. That's citrus. I grew up in that town. I grew up walking through citrus groves like that to school. In lots of places in the world, kids throw snowballs at each other. We threw oranges at each other. That's what we did. But they're not actually a great system to study. This is another one. Anybody know what this is? That is a really sad olive tree. Oh. That's what happens when you grow an olive tree in Palm Springs. If you don't know Palm Springs, it's a desert resort community. And I feel so sorry for that poor olive tree. Every time I see this picture, it must be a little bit embarrassed. Of course, then there's grapes, right? And grapes is, in my opinion, the perennial model system. And so it seemed to make sense. Maybe I should work in grapes. So what do we know about grapes? This is a figure that may be familiar to some in this room because it comes from Montpellier. This was my Bible when I started to read about grapes. It's a paper by Patrice Peace. And it shows some of the changes that have occurred during the domestication of grapes. So on the left, we have domesticated grapevine, Vitis vinifera. And on the right, we have the wild relative. You can see there's a change in leaf morphology, a very interesting change in mating system. There's differences in the size and the taste and everything about the berries. And then there's a difference in the size of seeds. And so what do we know about domestication, about grapes? Well, domesticated, give or take, 8,000 years ago. And maybe more than once. There was a science paper recently that seems to show that's the case, that it was domesticated twice. Like I told you, it's a perennial, and there's no evidence of a bottleneck effect. That is, genetic diversity in the domesticate is about the same as the genetic diversity in the wild relative. And there's all these phenotypic shifts that I've just told you about. Now, what about the wild relatives? What do we know about those? Vitis is a really interesting genus because there's about 70 species, 30 of which are in North America. Those 30 species are absolutely critically important because they are the basis for rootstocks. Over 90%, at least that's the estimate I have seen, of grapes that we grow for wine and for fruit have rootstocks from North America. So those species are really economically important. They are morphologically and ecologically diverse. And there's some really exciting evolutionary questions. I'm an evolutionary biologist. And what's exciting about them? Well, there's a few species that are purported to be the result of hybrid speciation. There's lots of introgression. They can all interbreed. And you can see from this map that they are found throughout the United States, including down at the bottom left where I'm from, where it's very hot, there's deserts. They're found in riparian areas, but they're heat resistant, drought tolerance, salt tolerance. All these very important traits can be found in these 30 species. And just to show you how diverse they can be. This is one of my favorite figures. I wish it was from my paper. It's not, but it's showing leaf morphologies of 8 different species in a Vitis from North America. Now, one of the great things about this system is that there's actually a living collection at UC Davis. And I'll talk more about UC Davis in a minute. And that collection has been sampled over 30 years from throughout the US Southwest. So Texas, California, Arizona, etc., which of course is very handy because we have our sampling done for us already. So I'm going to talk to you about 2 things today, all having to do with the genus Vitis. The first one is a little more genomic-y, and I'm going to talk about some of the genomics, some of the things we've found from studying the genomics of grape. And the other one is more kind of not ecological, but maybe we could call it ecological genomics-y. And we're going to talk about those wild relatives and where they are on a climate landscape. And I have to say this very carefully. I'm not a Vitis expert. I'm trying to learn. The experts are my co-PIs on this grant. Andy Walker, who's a breeder, he's just retired, which is sad because he knows a lot. And Dario Cantu, I'm sure many of you know him. He's just an absolutely great researcher and I love working with him. So everything I talk about today is in collaboration with these guys. Okay, so how did we start and meet Vitis? Well, we started very modestly. We sampled about 30 different. Dot represents something we've sampled. By sampling, I'm talking, whole genome resequencing. And we just did sort of a basic evolutionary study. So I don't want you to worry too much about the sampling. It's very modest and there's some table grapes and some wine grapes and we did a PCA and it doesn't matter. But what I do want you to see is this. If you measure nucleotide diversity in the cultivated versus the wild, you can see it's about the same. At least with this sample, there's no evidence of any bottleneck, which would mean reduced diversity. In fact, in some samples, cultivated actually seems to show more diversity than the wild, which, right, is a bit of a mystery. And you can look at heterozygous SNPs, and there the cultivated has a little bit higher diversity. Okay, whatever. These are just summaries of a data set of about 30 samples. We also did this thing that's kind of hip now, and that is we measured load. Now, how do you measure load? You bioinformatically predict deleterious mutations, and you count them within genomes. And so we counted all the deleterious mutations in the cultivated genomes and the wild genomes, and you can see there's many more in the cultivated genomes than in the wild ones. So, we thought that's a little weird, and that's especially true in the heterozygous state on the right. Okay, so there's more deleterious mutations in the clonal propagates. Okay, how do we explain that? Well, as population geneticists, we do what everybody does, and that is you just do simulations, right? So we did some simulations. This is forward simulations that are presented in sort of a weird way because the past is over here, the present is over here. So we're simulating populations. And you see there's 4 different colors. Those are 4 different populations that we're simulating. And the 2 that look very flat, that's outcrossing. So what we've done in these simulations is we introduce deleterious mutations, okay? And then we just see what happens to them. And we can measure fitness in these simulations. So you can see in outcrossing, there's 2 lines, one of which has additive deleterious mutations, meaning that when they're in a heterozygous state, they have an effect. Some that are recessive, but notice that really there's not much change over time in the fitness of those 2 outcrossing populations. What happens when you make something clonal? Well, that's kind of interesting because you can see there's 2 very divergent lines. The one here on the bottom, I'm colorblind, is a blue one. And purple. The purple one is when deleterious mutations are added. That means they have an effect on fitness in the heterozygous state. Once you become clonal, if they have an effect on fitness, boom, your fitness just decreases. But the really interesting phenomena is the one at the top, which I think is yellow or green. Green, okay, the green one. So we introduced a bottleneck and then we said, let's make everything clonal. And what happens? You introduce deleterious mutations. They're introduced as heterozygotes. They are recessive, so they have no fitness effect. And in fact, your fitness goes up. And why is that? Because you never have outcrossing to recombine and make the deleterious mutations homozygous. So, cool. This is sort of an explanation for why we might see more deleterious mutations. Now, we thought to ourselves, that's interesting. What does that mean for structural variants? These are things that change, what the genome looks like by losing or adding pieces of DNA. And we did this. This is an old paper now. We sequenced the Chardonnay genome. Now, 5 years later, I would say, what an embarrassment. We were very proud of that genome when we did it. Now, 5 years later, the technology has changed. That's probably a really horrible genome. But it was helpful for us because we could see things like this. This is a genome scan or a piece of the genome. And what you can see is those colors in the middle. Those are variant sites, SNPs. And you'll notice on either side of those colors, there really aren't any SNPs. And we were puzzled by this. And then you can also see coverage, right? You see there's the coverage, it's higher where there's SNPs, lower otherwise. What's going on here? Well, we realized a lot of the genes are in a haploid state. That is, there's been a structural variant such that one copy of the allele is gone. So, okay, that's not so surprising. What surprised us is when we estimated how many of them were haploid. I'm going to use the term hemizygous. Some people don't like my using that term, but I'm going to use it. I'm sorry. When we looked at how many genes were hemizygous, we were shocked. 1 in 7. About 5,000 genes in Chardonnay genome are hemizygous. You can imagine, I said to myself, this has to be wrong. And I went to my Drosophila friend down the hall, said, what do you think if I told you that 5,000 genes only have one allele? He'd say, you're absolutely dead wrong. But we're not. We tested it. We looked at it in various ways. We even had other people do it independently. Okay. So we have these clonal lineages like Chardonnay that have deleterious mutations and lots of structural variants. Now, do those structural variants mean anything? Do they have an effect? We know some of them do, because when we compared our Chardonnay genome on the left to Pinot Noir, actually it wasn't Pinot Noir, it's this reference genome, we saw that the blue slots there show an inversion on this particular chromosome. And we thought that was kind of interesting because this particular region of the chromosome had these genes called mid genes. Those are very famous genes because they regulate the process of anthocyanin biosynthesis. What does anthocyanin do? It makes purple grapes, right? Purple berries. So we thought to ourselves, what is going on in this region? Now it turns out the transition from purple to green berries is super well known. It's one of the, it's one of, like, in terms of connecting genotype to phenotype, it's one of the best known examples in the plant literature. And it turns out that there's this transposable element that knocks out one of the mid genes. Great. So you have to turn it off to get green berries. You also need another gene turned off. Okay. But remarkably, these two null mutations are found as heterozygous in lots of dark berried lines. So then when you get green berries, the question is, how do they become homozygous? Well, what we found studying Chardonnays, they don't become homozygous, they become hemizygous. Structural variants, and there's only one copy of those things. So we weren't the first to find this. I just want to point this out. It's a really beautiful study about a somatic mutation in Tempranillo grapes. And it turns out when they did some coverage statistics, they found that same region had been hemizygous. And we could look at the transition in evolutionary terms, how often you go from a dark grape to a green grape. Turns out that's happened a lot. Everybody knows that. And every time where we could get public data and try to look at that, we saw a coverage difference at this region. So it seems like becoming hemizygous certainly has a phenotypic effect. So then you might ask yourself, well, is this a property of grapes or is this a property of clonality? We've done the simulations. What's going on? So we have this paper that's not published yet, in which we got some really high quality genomes from some grapes, potato, cassava, apple, all of which are long-term clonal lineages. Then we got some outcrossing species to compare. We took some self-inbred species sort of as a control, because those should be completely homozygous. We should see no hemizygosity in those. And we used our methods, which is mapping reads back to the reference. And just quickly, here's what you see. This is the complete amount of the genome that's in a hemizygous state. So we have our clonal samples on the left, our outcrossing samples in the middle, and our inbred samples on the right. The inbred sample is cool because it shows that our type 1 error, our miscalling error, is less than 1%, which is nice. But you can see there's a difference. You can also do that just for genes, so that genome-wide included transposable elements and other things. If you do it just for genes, you see the same effect. Now, don't get too excited about this, because in some ways this is a horrible sample. They're not all independent points, but hopefully you're getting the point. When you have a clonal species, it accumulates all these mutations through ratchet-like mechanisms. By the way, not all clonal species show this. We did a study of avocado, sequenced an avocado genome, and it didn't have a lot of hemizygosity. And we were kind of puzzled, like, avocados are clonal, shouldn't we see this? Turned out the tree we sequenced had only been clonal for 2 generations, so like for 20 years or something. Okay. By the way, this work is done in collaboration with Young Feng, who was a postdoc in my lab when we started. Great work. He's now in Shenzhen. He has more money than God himself. I just talked to him the other day. He has 61 people in his lab. Can you imagine that? So, wow. So which genes are hemizygous? I've already showed you there can be phenotypic effects. That's the berry color thing. But we can actually kind of guess what genes must be hemizygous, because if you want to keep fitness high, you have to have things. This was done on SNPs that are deleterious recessives, but you must probably have structural variants that don't have a lot of effect on fitness. So you might imagine the hemizygous genes probably don't do very much. And we've looked into that with this data set that I just described to you. And rather than give you the details, I just want to give you the answers. Hemizygous genes are smaller than the average gene. They evolve more quickly. They are less broadly expressed and less often expressed. But interesting, their methylation patterns make them look like they're functional genes. So some of them are probably functional, but a high proportion are probably pseudogene-like. Could they be misannotated genes? Doubtful, because a lot of them are expressed. Interestingly, we could also look at hemizygous TEs, and that story is actually a little more interesting, and I don't have time to tell you about it. But what's interesting is hemizygous versus diploid TEs, right? Just one copy on one of the 2 haplotypes of the genome. They're more extensively methylated than the diploid ones, and they have large effects on the expression of nearby genes. So probably these hemizygous regions, in terms of TEs, do have an effect subtle on phenotype. Okay, so I started with this mystery, and I haven't answered the question for you yet, and I can't. But why are perennials, why don't they show evidence of a bottleneck during domestication? We know there are some things. Oh, here's this graph I showed you. No evidence of a bottleneck, no reduction of genetic diversity. So how can this be? Well, we know there are some things that usually didn't go into the simple models of domestication, like introgression with wild relatives after domestication. That's a growing field where we're realizing crops got domesticated and then they're being moved to new locations, and there's introgression with wild relatives in those new locations that increases genetic diversity. There's multiple domestication events. I talked to you about grapes, probably domesticated twice. They came from 2 different populations of wild relatives. But I'd like to suggest to you that also we're probably being misled, because these clonal lineages, after all, perennial crops tend to be clonal, are accumulating in a ratchet-like mechanism, lots of genetic diversity. And so I haven't studied this yet. This is just a suggestion. Anybody wants to study it, I'd be happy to study it with you. But my guess is if we modeled this properly, that a lot of the genetic diversity we're seeing on the cultivated side is one, heterozygous, and two, probably happened after domestication. So by just comparing these two things, we're kind of misleading ourselves. So that's the first part of this talk. Oh boy, oh, I'm good. So summary, hemizygosity. Clonal propagation leads to the accumulation of recessive deleterious variants, lots of SNPs, lots of S structural variants. As a result of this process, about 13% of genes across different crops, clonally propagated crop, are hemizygous. Now that number 13% is going to depend on how old the clonal system is, right? Hemizygosity has the capacity to drive phenotypic change. We've shown that in the case of berry color. But most hemizygous genes are small, kind of, they're worthless genes. Can we say that? They're worthless. Is that politically correct? All right. So that's the first story. Let's turn to the second story. That one's done. And we're going to change our viewpoint quite a bit. We're still going to work in the genus Vitis, but now we're going to look more about the wild relatives of the crop. So you all know this, but I think it's always worth saying. The crop wild relatives of any crop are critically important because they are sources for improvement of the crops that we rely on. Things like cytomesic male sterility, resistance to pathogens, drought tolerance, salinity tolerance, you name it, breeders rely on the crop wild relatives. And it has a huge economic impact, $186.3 billion a year. I have to tell you, I have no idea how they come up with that number. But it's an impressive number. That means that there has to be and should be concerns about the conservation of these wild species, both in germplasm banks and then in the field, right? And they are critically important for things like this. There is the strategy that's been called FIGS, Focused Identification of Germplasm Strategy. And the idea of FIGS is to include climate in how you look at crop wild relatives. So let me give you an example. That's the easiest way to do it. This is a map from a paper that's from Carl Schmid's lab. Carl is in Germany, and he's trying to breed soybean that does well in the winters in Germany or overwinters or with this, whatever, the spring, right? In the climate of Germany. So if you go to germplasm banks of soybean, there's probably 10,000 accessions. Which of those accessions will be most helpful to you as a breeder in Germany? Well, what you want to do is you want to find accessions that are from the climate that's most similar to Germany, right? Because it's already pre-adapted to what you're trying to do. So this map is supposed to be something about climate, and all those little dots represent places where Carl thought the accessions from there, both wild and cultivated, might be helpful for his breeding program, right? So it's using climate to help filter and define the right materials for your breeding program. That's great. Now, of course, you can't really these days just breed for, say, Central Germany in 2024. You really have to breed for Central Germany in 2050, right? Because the climate is changing so rapidly. So in addition to wanting to look at the current climate, you want to look at predicted climates and do your best guess. Making sense for everybody? Few people are saying yes. Thank you. Okay, so how do you look at climate? This is stuff I think probably everyone in this room knows. You build species distribution models. That's been the main tool to look at climate and how it relates to a species. And it's actually a really simple concept that's a little harder in detail. So the first thing you do is map the species in the wild. What do I mean by that? You find out where it is located in the wild. And then you measure these things called bioclimatic variables, things like how much rainfall, how hot does it get, what time of the year is it hot, when does it rain? And then you project where the species lives in niche space. So here's an example. This is a wild relative of corn. Great. When you look at where it's located, this is supposed to be a map of Mexico. Where it is located, you can find out the climate variation in that location and then project where you think you could find this species. So those blue and red parts are where we think we can find these species. By the way, usually you would use one color because you're only studying one species. We actually purposefully split this into two colors because there's some warmer weather populations in red and colder weather populations in blue. Cool. This is where you expect to see it. Now, what do you expect to see if you take a climate model that models what's going to happen in these bioclimatic variables, and then you project that niche in the future? This is what you see. Great. Now, there's some good news and some bad news here. The good news is there's more red here than there is there. So you might be tempted to say, hey, that's really great news for ZMA subspecies mexicana. But you can see there's almost no blue. So these cold weather populations are in trouble. They're at higher altitude and their niche is going to change. But there's also some bad news in the red part because you can see it's disjunct. And so you have to ask, will ZMA subspecies mexicana be able to move to those locations? We don't know. So this has been done on lots of things, including lots of crop wild relatives. But there's a problem. These things ignore the potential for plants to evolve, which we know happens. So how do we include evolution into these models, thinking about climate prediction, crop wild relatives? So how do we do both? That is, which wild populations are well situated for adaptation, conservation, and which are best suited to contribute to breeding, sort of in that, like that figs idea I told you about. Well, there's a way to do this, and I wish it was my idea, but it's not. Let's go out and sequence those populations. So now I'm saying, hey, we sequence the red populations, we sequence the blue populations, and we can see, hey, there's at this particular sites, could be rad sequencing or whole genome sequencing. This particular site, the reds all have A, the blues mostly have G. What would you, as a population geneticist, what would you conclude from that? Well, there's a few things you could conclude. One is that maybe these populations are historically separated, and so they've had time to become genetically distinct. Could be. But what if I told you most SNPs look like this? So we have genome-wide SNPs, and most of them look like this, where the T's and C's are shared between the two populations. So the genomic background isn't that there's been genetic differentiation of these populations, something else is happening. And in that case, you might say to yourself, well, this locus that we started with is an outlier. It's not like the rest of the genome. Great. Well, you can do genome environment associations and ask yourself, are those SNPs associated with environmental variables? Rainfall, heat, whatever, right? And these are, well, you probably all know about these methods. Okay, cool. All right. And I just want to say I made multiple circles to show that really what you want to do is sample multiple populations. So these are candidate loci for environmental adaptation. Great. What do you do with that? You find 50 SNPs that are candidates for environmental adaptation. How can that help us understand what's going to happen in terms of climate change and in terms of adaptation? This is the really clever idea. By a paper, if you haven't read it, I highly recommend it. This Ecology Letters paper. Fitzpatrick and Keller came up with this method that I'm just going to give you a brief overview on, that I think is really fantastic. It's been used a lot. It has problems, but it's great. And I want to explain to you what it does. So we're going to spend a little while on this graph. Okay, what you do is, let's say we find 50 candidate loci. Great. What do you do then? Well, you want to know how they're associated with climate and how they're going to change, or what that tells us about predicted climates. How do you do that? Well, you take those 50 SNPs and you put them in something called gradient forest. It's a machine learning. It's basically random forest regression with a few changes. And what it can do is identify nonlinear associations between climate variables and genetic variation. So let's say our climate variable is temperature, And on the y-axis, we have something called the allelic turnover. You might want to think of this as changes in allele frequencies for populations. Okay, great. You can get a nonlinear. This method will identify the important bioclimatic variables. Is it rain? Is it temperature? That's great. And the SNPs that are associated. So you start with some candidate loci and you find only a subset really associate with different bioclimatic variables. Great. What that does is it produces a cumulative turnover function. And that's what this graph is. And this graph was taken from another paper. So what is this graph telling us? Let's imagine we have population A and population B. They are in different places in climatic space, that's the x-axis. And for those alleles that we're looking at, or those loci, say our 50 SNPs, they also have very different allele frequencies. B has this one and A has that one. Just think of it as, yeah, allelic change. Great. Why is that helpful to us? Because we can also predict climate. When we predict climate, and in this particular example, it's really nice. Let's say we're predicting the climate in 2100. And we say, well, population A, if it stays in that population place, it's going to change that much in its climate. Population B is going to change the same amount. Their x-axes, the CAA and CBB, are the same. So these two populations are going to change in their climate. Great. But what does that mean in terms of how much allele frequency change you need? That's where this linear curve is super helpful. Because if you go from here to A prime, on the y-axis, that's a huge amount. It's saying this population is, in terms of evolution, it's got to evolve more. Its alleles have to change more than this one, even though those two populations are undergoing the same amount of temperature change. Hopefully this is making sense. This thing here is called the genetic offset, the distance on the y-axis. Now, this is done in 2D space, but you can do this across all climates and all SNPs. It gives you a measure of how much change has to occur. So here's an example. Going back to Zea mays mexicana, here you have all the bioclimatic variables, those things I didn't label them for you. Some of them are temperature, some of them are rainfall. It shows you which ones are the most important, and those are the colored in ones. And then you can build a turnover function. And this part is just like what I showed you. And by the way, these are our warm weather populations and our colder weather populations. And we've drawn it for mean temperature in the driest quarter. Cool. These lines are individual SNPs. So you can look at individual SNPs and say, how much is allele frequency going to have to change in individual SNPs? Down here, the dotted line are background SNPs, right? So we get a very different, it's just sort of a control. Okay. And then you can ask, when their population changes, how much is the change on the x-axis or the y-axis? And the cold weather populations are going to have to change more than the warm weather ones. Okay. So we have this way to sort of guesstimate how much evolution has to occur at a putatively adaptive SNPs in wild populations. So how do you apply that to wild relatives of Vitis? I just want to say, we're interested in lots of questions in this group, such as species relationships and introgression, sex determination. It's a really fun topic. Even pangenomes. Daria's group just published this wonderful pangenome, reference-free, which is kind of rare in plants. And those are the people who did it, both French, by the way. Okay, coming back to this idea, but we're also interested in climate. So here we have a graph. We did this sort of, I would almost call it a pilot study to look at these methods in this group. And we looked at 5 different species, and here we have their species distribution map on the United States. And we could look from the pleasant to predicted climate in 2050, 2070, 2090, and we'd ask ourselves, is the population where we sampled these things, is it expected to be okay? to still be within the species distribution map. And you can see some of these, like Vitis gerdiana, there's a lot of white at the bottom there saying, those populations are in trouble in terms of species distribution mapping. And then you can measure offsets, this thing I just talked about using gradient forest. And you have to be careful, you can't really compare across species, but you can see that within gerdiana, there's some variation and some of these populations have higher offsets. Based on this method, we would conclude they're in more conservation danger. We can also do things like this. I would call this a FIGS approach using genomics. So what we have here is a map of the United States. This is not a political map. I know usually when you see red on a map, in fact, I just want to point out, the other day, this would have been blue, and that's where I'm from, okay? All right, but what this map is kind of interesting, it's kind of fun. We went to this database called GBIF, and the circles represent places where grapes are grown as a crop. Is it complete? No, because some people grow grapes in Texas and blah, blah, blah. But it shows us sort of a predominantly where grapes are grown. And you can see there's a lot in California. The red represents predicted climate for grapes. Red means that when you are no longer in the red, that's not a good thing. That means that grapes will no longer be able to grow there, right? Based on these species distribution models. Okay, that's interesting. What can we do with it? Well, we had all these wild relatives that we measured genomic offsets with. So for example, we could take a wild relative from Texas and ask ourselves, where will it have a really low genetic offset in the future based on future climates in a way that might actually help with grape breeding? I mean, grape, viticulture. So it's essentially asking, let's take this here, predict the climate here, and ask whether there's a large genetic offset. Does it seem genetically adaptive to be adapted to grow there? And I don't want to go into this in great detail, but I do want to say this wasn't my idea either. This actually comes from Montpellier as well, the Vigorous group. They published a great paper a few years ago about pearl millet doing the same thing in Africa. So what we did find was kind of fun, that the best species we could find that seemed to most adapted to help, especially in this region where the dark circle darks are, in the year 2070, is this thing called Vitis mustangensis. Now, the reason this is interesting is it's from Texas, it's drought tolerance, it's salt tolerance, it has all these things, but to my knowledge, it's never been used as a rootstock. Now, will it be a good rootstock? Who knows? It may not graft well. It may affect the taste of the berries, but it's an interesting way to sort of predict and maybe focus on something that might be of breeding potential. And I call this FIGS on steroids. We're doing this FIGS approach like Carl Schmidt did in soybean, and now we're adding genetic information. Okay, but we can also do this method with traits. And I think this is the first time it is ever done. We were interested, well, Andy Walker traditionally has been interested in something called Pierce's disease. It's a bacterium, Xylella fastidiosa, which interestingly enough, was first identified as a problem in grapes about 20 minutes from me right next to Disneyland. But that was in the 1860s. Now it's a huge economic problem in grapes, but not only in grapes, you've probably heard about Xylella because it also affects almonds, it affects olives. So you've heard about probably the problem with olive trees, particularly in Italy. This bacterium has made its way to Europe from the New World and is causing all kinds of damage on lots of different crops, crop loss across grapevines, almonds, olives, etc. The interesting thing is none of the crops have resistance to this pathogen, but what does? The wild relatives, the Vitis wild relatives. And we want to study more in depth resistance to this thing. And then we want to use that information to sort of do some climate projections and thought. So how did we start? Well, the first thing you do is you phenotype resistance. This is like a miracle to me, not the kind of thing I do, but Andy and his co-author Sumaira did these assays where you inject the bacterium into the plant in the greenhouse. So you have cuttings of all these different plants. You go to the Southwest collection. They did this for hundreds of plants. You let them grow for 10 or 14 weeks. You do all the right controls. You do randomized designs. You measure the plaque density of the bacterium in the plant, and then you get yourself a phenotype for each plant. So that was a ton of work taking lots of years. But in the end, we had, for example, in this one species, Vitis erizonica, we had about 200 plants that had been phenotyped for resistance to this important bacterium that affects lots of crops. And if we put that on a map, you can kind of see it. And essentially, we have a quantitative measure of how much bacteria there is. And we just colored the dots where we had sampled those plants and how much bacteria grows in them when you infect them. So this was work done by Abraham, a postdoc in my lab. And so one thing you can do, of course, is a GWAS. So I think probably everyone in this room has had a paper like this one, where you really love it, but it seems like nobody else does. And I think the problem with this paper was, here's a GWAS, and everyone says, GWASs have been done on plants and resistance to pathogens all the time. That's what the reviewers told us. Oh, this is not interesting. But I have to tell you something. It is interesting for a few reasons. First of all, if you look in the literature, there's very little GWAS work done on wild plants other than Arabidopsis and maybe some brassicas. So like to do a crop relative, is really substantive in my opinion. The reviewers weren't convinced. The other thing is, well, we looked at GWAS, we used all different kinds of methods, including some SVs, some structural variants. I don't care so much about that, but we found this peak here. Well, that's a known peak. Andy Walker in years past had done very low resolution QTL studies and found that there's resistance on this chromosome. And in fact, he's actually back-crossed that resistance into some great varieties. But there's also other regions of the genome that have peaks. You always see that in GWAS, right? And SNPs that are associated with the phenotype. Now remember those SNPs, because we're going to use those in just a minute. Just want to say, by the way, we did some functional work. This is from Dario's lab. We're, complimented. Still, the reviewers didn't like it. But for the first time, we did some expression, and we now have some really viable candidate genes for resistance that are being tested functionally. And if those work, they could be really useful in bunches of crops. Okay. I'm a population geneticist. I like to look at patterns of diversity, and we saw some really cool stuff. This is that chromosome 14, where there is this region that we know imparts resistance. And then there's another region that also is a GWAS peak, and those two regions are in linkage disequilibrium with each other. I don't know what that means. It might mean that in practice, these two regions are being selected together, or maybe they're synergistic in some way. But I'm not sure I've ever seen a plot quite like this one. And just to show you, here's the GWAS peaks, and those two squares are in linkage disequilibrium with each other. Bizarre. Okay, now let's get back to this gradient forest idea. We have SNPs that are associated with the trait. Why don't we just study those SNPs on the climate landscape using these methods? Okay, so we just said, why not? Nobody's ever done this. Let's take trait-associated SNPs. It's a little bit like genomic selection. You take them, and now we're just going to put them in that gradient forest machinery, and you get things like this. Well, here's the bioclimatic variables, and there's their importance. So this is different than the other graph I showed you. It's flipped, but who cares? There's one thing that really is always the most important predictor of the SNPs associated with resistance. It's this thing called bio8, the mean temperature of the wedge quarter. What does that mean biologically? I don't know. But now we have a variable that's related to resistance. and it's a climate variable. We can do other things. You can say, okay, can we predict resistance based on different kinds of SNPs and stuff? So for example, if we take the SNPs that are associated from the GWAS and say, are they associated, how well are they associated with resistance, the quantitative trait? It's pretty high. You'd expect it to be. That's a circular thing. You find the SNPs and then you find, right? But crazily enough, if you look at Bio8, it actually measures resistance quite nicely. So in this system, you can almost just measure where they come from and predict whether a plant is or is not resistant. Kind of fun. But here's where it's more fun. Because when you look at the various plants, that's all the individual circles, and you build this turnover function, you can see it's very steep and that there is basically an inflection point somewhere in 7 to 10 degrees. Above those temperatures, most are colored in, meaning they are resistant. Those plants are resistant at those temperatures. So there's the inflection point. There's the resistance. And most of the ones at low temperatures are susceptible. Kind of neat. Why is that important? Because we thought, remember, this bacterium infects lots of crops. So we said to ourselves, is this something having to do with if you are in low temperature, you never see the bacterium and you're never infected? So we went to a global database of crop infections with Xylella fastidiosa. And I don't know why it looks bimodal like this, but the main thing is underneath 10 degrees, you rarely see infection by this bacterium. So what we're learning here is that by studying resistance in the plant, we're learning something about where the bacterium, we think, is. Or maybe it's the insect vector. We don't know what, but it's telling us something kind of neat. Why is that neat? Because then we know it's associated with Bio8. We can predict Bio8. There's climate models that predict it. And we can ask ourselves, where in the world are we growing things where Bio8 is going to, in the future, become more than 10 degrees, even if it's currently less than 10 degrees, right? And why would we do that? Because when that climate changes, we expect when it gets over 10 degrees, that might be a place where this bacterium is happy. Hopefully that makes sense. So what is this graph showing us? Everywhere that's, I think, red, including parts of France, that's where we expect this Bio8 to increase to be over 10 degrees, even though it's below 10 degrees now. And what does that mean? From our perspective, it means that there's going to be increased pathogen pressure. Make sense? The blue areas are actually where the reverse is happening. We're not worried about those. So you can say, okay, where are grapevines grown? Where are almonds grown? How much of the growing region of these various things are going to be affected? And we estimate about 40% of where grapevines are going to be have increased pathogen pressure. 18% of olive groves, big concern, 5% of citrus groves. Okay, you guys look very tired. So I better quit. I just want to say there's problems with this method, right? You're using, sometimes machine learning is like a black box. Also, you're focusing on a very small number of SNPs. I think in the trait case, that's great. We have a small number of SNPs, but if you're doing this on a population scale, you're ignoring most genetic diversity. You're ignoring genetic load, deleterious variants. That has an effect on fitness. So how can we improve our modeling to include not only adaptive SNPs, but deleterious SNPs? Consider things like gene flow, dispersal, etc. The answer is, I don't know, and I'm going to skip this. The answer is, I don't know, right? These are early days for these kind of models. And I feel there's this allegory of the cave with, is it Socrates or, Socrates or Aristotle, one of them? Socrates, thank you. I'm glad you have a classical education. And in that allegory of the cave, people are in the cave and they're looking at shadows on the wall and they think that's reality because they don't get to see reality. And And sometimes I feel like these models are like that, and this is true of all population genetics. You're seeing the outcome of your modeling or whatever, and you're not really sure it's true. So are these predictions based on gradient forest real or not? We're not sure. But there's some things we have to do yet. One, we've been working on these problems in the wild relatives. Two, we need better formal quantitative models that consider not just adaptive SNPs, locally adaptive SNPs, but things like adaptation, migration, base load. Three, believe it or not, there's no real practical guidance on how to use these methods. They're being used a lot now. That's just an advertisement for, we just did a simple subsampling meta-analysis to try to get at best practices. But the most important thing is we need to ground truth these predictions. That means growing plants in climates where we can really say, we don't expect these to survive based on our gradient forests, and do they survive? Or what really happens in reality? And we're actually trying very hard to get funded to do some ground truthing, looking at drought in the greenhouse with the Southwest collection. What we're hoping to do is really connect phenotypes to some of these predictions in an agronomically important trait, but also a complex trait, more complex than resistance. Okay, so that is mercifully the end of my talk. I have a very old picture of the Gaut lab here because you might know somebody. First of all, yeah, unfortunately, this looks like the Last Supper. It was not. There's Aline, who was in my lab for how many years? 3 years. It was great 3 years. No, a lot of it was the pandemic, so some of it wasn't so great. There's the Cantu lab, and I've mentioned Andy. Now, I want to say one more thing before we have questions, and that is, Aline mentioned I'm the editor-in-chief of MBE. One thing I have always really valued about the French scientific community is that I think more than many other places, it values scientific societies and society publishing. And I am nonetheless always amazed at how many, especially students and young researchers, don't understand how important society-based journals are. And for MBE, the money that you pay for open access is moderate compared to something like Nature Communications, right? So it's affordable. And that money, most of it, goes back to the society. And what happens to that money? Well, Aline knows better than most because she's the treasurer for the society. But that money goes for sponsoring the annual conference, sponsoring symposia, not just the annual conference, sponsoring graduate fellowships. So it's one of these things where it's run purely volunteer. All the associate editors are practicing scientists. They're not these people who can't make decisions. Anybody who nods has been to Nature before, right? I hate the editors there. Don't tell them I said that. And I would just urge you to send your best work to MBE or to Plant Cell or to Genetics or G3. Anything that's society-based, it's bang for the buck, and I think it's a better use of the public dole because it is public money that we have to pay for these things. Okay, that's that.
So much. We have time for questions. I saw quite a few questions online already. Is there any question in the audience? Yeah, I can start with. So, yes, there's one. Yeah, yes, please.
Yeah, I was wondering in the first part, you interpreted this hemizygous as mostly being caused by kind of ratchet effects or basically they increase. Yeah. Don't you think they could also have contributed a lot to adaptation or selection under domestication just because everything has to remain so heterozygous in those clonal? Yeah, is that sometimes something that's recessive?
I sort of overplayed it a little bit, but that's why I talked about the berries, right? Berries are under selection, and so you get this hemizygosity that's selected, the green berry color. And I think
a new way to figure out which either of these are kind of drift flow basically, as opposed to really things of the red.
Yeah, you mean for particular loci.
Or the proportion of hemizygous sites that are driven by adaptation.
Haven't thought about that. I mean, we've done Go annotation and there's some enrichment, but I don't always believe those, so I didn't want to talk about that. But yeah, you're talking about something more like,
Selection.
yeah. No, we could do simulations to get at that and we just haven't done it, right? Yeah. That's a great idea. There's too much to do.
Because I have this impression that, it seems from several of these types of studies, even for duplication and things, that they occur a lot, and whether they spread or not might depend on whether this selection does not necessarily, doesn't have to be that relaxed, purified selection. It could actually be positive selection.
Yeah, absolutely, especially in a cultivar. I think probably we don't know what percentage of those homozygous genes actually contribute to things like
Traits.
traits, pace variation, for example. We just don't know, but certainly some do. So, thank you. Yeah. Should think about that.
Oh, it was great, beautiful. Thank you.
Oh, you're welcome. Thank you.
Just one idea, because we tested some of these amazygosity condition, and I found out that it could also be, I cannot quantify yet because they are still working.
Okay.
But there is a phenomenon of cryptic parentage. Cryptic parentage is in one portion of genome. You have a common ancestor long time ago. But if you calculate parentage on the whole genome, you don't see it because maybe it happened 4 or 5 generations before.
Right.
So you cannot detect normally. But in this region, they have the same allele from different ancestors.
Okay.
So they are not homozygous, but they are homozygous.
But I guess I'm not following because you still have these differences in copy number.
No, but they were the two parents 3 or 4 generations ago. They were parented.
Oh, I see. So you're
They
saying,
have the same chromosome, the same.
you're saying it's not a product of clonality and mutation, that it's rather.
It's an ancestral.
That's entirely possible. But I think that doesn't affect the estimate of how many are,
No, sure.
copy number, right?
Yeah.
I buy that for sure. But at some point in the lineage, there's been mutation.
But it opened the question: how comes there are some stretch of haplotypes that are conserved actually along so long time? They must be advantageous.
Yeah, I think you're right. Housekeeping genes. Oh, many questions.
Also about this hemizygous gene.
Yeah.
Do you see differences in the number of hemizygous genes between the species that are more used for rootstock, so they don't need to preserve the functions of the ground part?
Yeah, so the rootstock, that's a great question.
I don't know if it's relevant compared to the rights of the
Actually,
rootstocking.
I might need a Vitis expert to help me with this, but so we measured some of the rootstocks, but those are all outcrossing, right? So they were more like a comparison. But at some level, they are also clonal, I think. And that, I don't know how long they've been clonal. I mean, rootstocks just started in the 1860s. So it's much shorter than, Cabernet's like hundreds of years old. So it's a much shorter amount of time, but yeah, good point.
Yes, thanks for your talk. I have a question regarding the second part of your talk, where you showed a graph showing the genomic offset for different Vitis species.
Yes.
Remember this one?
Yeah.
And you had for some species quite large variation of genomic offset.
Yes.
Is it only linked to the range of geographic distribution, or do you have any other interpretation explaining these differences in variation?
No,
between
that's
species.
that's a great question. So like Irvine, we showed had a bigger offset. That's right.
Yeah.
My recollection, and I'd have to look it up, is that it's actually a more tightly, it's not spread widely. Like you saw Arizona, because it's across the whole state of Arizona and huge. I think Irvine is more restricted. I've always interpreted that as, despite that geographic restriction, when climate changes, the niche is changing more. So that means the niche is moving and there's a bunch of individuals that are going to be no longer in the right niche. right? If they don't move with it. Does that make sense? It has to do with but you're asking a good question. Is it just geographic space?
Yes, and then.
Yeah. So we're actually looking, I didn't have time to talk about this, but we're actually looking at in Arizonica. It turns out that a lot of the species in the north have dispersed over the last, since the, what is it, Holocene? And those have very different genetic signals than the other ones. And so we're kind of looking at that question, but I can't answer it for Gertie Ann. Yeah. Any other questions?
Maybe I had one again
Okay.
on the hemizygosity about the transposable elements that were deleted. They are the ones that have the strongest effect on nearby genes.
Yes.
And so it's similar to Louis' question, but is it selected, this hemizygosity?
So I think you have to be careful with TEs. I like to think for genes, ancestrally, they were diploid and you've lost a copy. For TEs, it could be they're new. And that's my guess is they've jumped, they're hemizygous, they're new, and so the host response against them is really strong. And so it affects not only their chromatin status, but also nearby genes.
So you would have to separate between the new ones and the older ones.
Yeah,
Maybe.
and we did, the classic, and they're newer.
Okay.
So that's, I think that's the right.
Okay, makes sense.
On average, yeah.
Thank you.
Sure. Yes.
What's your feeling about applying genomic offset studies on cultivated grapes? What would be the problems?
I mean, you can do it. You can do it. And maybe it would be a nice way to predict what cultivars should be grown where. Right? And so I think you could, I think it'd be fine. In fact, I'm thinking of Eve's paper on pearl millet in Africa, and essentially he did that. So I think I'd say go for it. Yeah. I mean, all these things have problems. The predicted climate models we're not sure about, right? The model itself, how good is it? But I think it's worth doing.
So sorry again
Oh my gosh.
about this. So actually, these are just deletions, right? That happen in the clonal.
Yeah.
They maintain. So do you know how this compared to the rate of deletions that occur in a more general setting, not non-clonal population?
No, we
There are
don't.
deletions that can't fix, basically.
Yeah, that's exactly right. No, we don't. And we're sort of at that stage where it's more a phenomenon than getting at the good theory behind it and, right, like mutation rates. I was thinking about this the other day. If you really want to explain why, back to this bottleneck problem, if you really have all these new mutations since domestication, you need to know mutation rates, the age of clonal lineages, like there's all these parameters that have to go into this stuff, and we don't know any of that yet. Does that answer your question? Inadequately, but yeah.
We have an online question.
Oh,
So we have a question by Marion Sinclair Waters. You can turn the mic on to listen to you.
Yes, thank you. Thanks for the talk. It was nice. I was wondering about where you, on chromosome 14, where you have those 2 blocks in high LD. Perhaps I missed it, but what are your hypotheses for why this pattern may be? And I was also wondering, can you rule out an assembly error?
oh, can you rule out an assembly error? I would make you talk to Dario about that. No, that's a joke. Can we rule out? I'd be very surprised if it's assembly error problem, because we've now sequenced several wild Vitis, and they're all very much, independently, and they're all very much in synteny. So I don't think that's the explanation. I don't really know what's going on there. There are some studies that have shown synergistic interactions in disease defense. There was a paper from that Lefeigel, and there was some sort of unknown association between alleles at one locus and another. And if you had the wrong alleles, then actually resistance didn't work at all. And so there's some sort of selection for having the right alleles, and they're sort of in that way linked, even though there's lots of recombination between them. So I'm guessing it's something like that, but we don't know the mechanism. Does that help?
Yeah, that's cool. Thanks.
Yeah, no worries.
Yes.
Well, yes, I was kind of surprised by the amount of hemizygous genes, also the outcrossing species.
Yeah.
It's actually quite close to what you see in the clonal.
Yeah,
So
it's about half. Well, yeah, close to half. Yeah, I think there's just a lot more structural variance than we imagined. I don't have a great explanation. Or are these things, you know, the really boring explanation is they're annotated as genes and they don't do anything. But I don't think that's fully it. So, yeah, I don't have an explanation.
So do you have any idea about how different these are between the lines, the different
No, that would be a great,
cultivars?
no, right, that'd be a great thing to do. Yeah, you mean, we could take the clones and look at whether they're different. We'd want to make sure we choose, I mean, I wasn't super happy with the sampling of what we've done so far. In that case, we'd want to make sure we choose clones that have been distinct for a long time and maybe a range, and then we could do that. We could do exactly that. That's a great idea. Young Feng with his 60 people, infinite money, I think has almost an infinite number of genomes. And now all the data are there, you just need to ask these good questions. Yeah.
Maybe one last question before lunch.
I have a question. Isn't there a tension in between the value of heterosis, and because if grape is so diverse and heterozygous, there must be a value of heterosis somewhere. But in this cultivar, you have a hemizygosity, so this goes in the other direction. So
That's right.
How do you?
I don't know.
No, we have no idea. Okay.
Yeah, no idea. You know, this could I think this could contribute to, well, this deleterious recessive effect. Probably this hemizygosity contributes to inbreeding depression.
Or maybe this kind of you call it the managed gene, the genes that are quite but housekeeping or something. So there can be a difference between the regions.
Yeah, my naive idea is that housekeeping genes for me. Typically.
Well, thanks again, Brian. It was a great seminar. Lunch or something later, let me know. And also Brandon is looking for postdocs, and I only suggest you talk to him if you're interested. He's the best mentor ever.
Oh, well, thank you.
When I retire, I will come.
Okay. The weather's good in Irvine. Here.
In 1 year and a half, I think it's so I'm sure.
Yeah, especially if we get